Tuberculosis or TB is a chronic respiratory disease that has plagued mankind for centuries. The first evident case of TB was reported about 3,000 years ago, which was found documented on clay tablets in Mesopotamia. It read, "What he coughs up is thick and bloody. His breathing sounds like a flute, and his hand is cold." Centuries after centuries, TB persisted and was called a variety of names, such as White Death and graveyard cough. A vaccine and 3 drugs later, TB is still ranked high on the world's deadliest diseases. The threat posed by TB is its drug-resistant nature, which urged the World Health Organization to declare TB as a global emergency. This makes us wonder, what makes this bacterium so resilient that it has stood the test of time? The answer is its tough surface structure. The structure acts like a fortress that surrounds the bacteria, giving it strength and protection against various antibiotic insults. The focus of my PhD is to understand the surface structure and find ways to target it. Previous research has indicated that the bacterium has a specific region called the intracellular membrane domain, or the IMD, as seen on the left-hand side of the slide. The IMD is enriched with many proteins that facilitate the formation of this tough surface structure. The IMD can be compared to a construction zone where various building blocks, the precursors, come together to form the fortress. The careful orchestration of proteins to further reactions can be compared to a mechanical crane that facilitates the building process. To stop the synthesis of the surface structure, we used a chemical inhibitor that decreased the levels of precursors entering the IMD. This can be compared to a roadblock that stops the building blocks from entering the construction zone. Altogether, the roadblock stops the building process and weakens the fortress. Our results indicate exactly that. The chemical inhibitor successfully disrupted the functioning of the IMD and halted many important processes. The bacterial cell, which is typically rod-shaped, started ballooning after exposure to the inhibitor, as depicted through the pictures in the center of the slide. The ballooning deformity implied surface structure damage, which eventually led to cell death. With this study, I strive to understand the mechanism of surface structure formation, determine its vulnerabilities, and help design effective drugs against this deadly pathogen. It's high time we eradicate tuberculosis. Thank you.